Hello, in this video we're going to be looking at the experiments part 2, so let's not waste any of your time. Let's get right to it.
It's hard to breathe, but that's alright. Hush.
Here, sorry about that, slight adjustment to the camera. I didn't double check that before I clicked go. Again, we're walking through a historical look at a variety of experiments conducted by a variety of scientists, all with the idea of understanding what is DNA. And as you know from watching the first video, I didn't do very well keeping up to my promise or my goal of matching what I talk about with the steps that the text has. But I will continue hopefully to improve on that. All right, so our next scientist, Rosalind Franklin, Maurice Wilkins. This was a huge event in the world of science, because we kind of moved from learning about or recognizing the DNA's existence to now starting to study its structure. And our journey ends by talking about a scientist who developed a process that really was a big part of the Human Genome Project, but we'll talk about that, Mr. Sanger. But this group here, where they begin to look at the structure of DNA and what exactly was this structure. And you can see there, using X-ray technology, they were able to essentially take a picture of DNA to try to understand what it looked like and everything. And their contribution to this was to learn that DNA again was a helix. Okay, it was a spiral, that spiral staircase shape. Okay, so our next set, Watson and Crick, they built on the information that Franklin and Wilkins provided. The idea of, okay, it's a helix, what's it made out of? And they were able to study and look at it, again, using X-ray process to kind of see, okay, what's it built out of? And their contribution to this was to learn that DNA structure, and now we know it pretty well. Even back in chapter 3, macromolecules, it's the idea of that phosphate, sugar, side rails, and in the nucleotides going along the inside, the ATCG complementary base pairs. Excuse me. All right, our next duo, Michelson and Stahl, they also reached the same conclusion. Instead of using X-ray technology, they again went back into the lab, and their advancement on this process of learning DNA was the idea of how it replicates. So we're going to look at their experiment. All right, in their experiment, they were studying DNA replication, and essentially what they worked out is how it replicates. So they took, as you can see, they have their first step was to some grow bacteria in a heavy medium and in a light medium. And the purpose of this, again, so they can trace where this material goes to. So that was their first step. Excuse me. And then their second step was to take that bacteria, put it in test tubes, and let it grow and develop new bacteria strains. And then, but with the bacteria that grew only in the heavy medium, okay, the only DNA they got was that same type, and DNA developed in a heavy medium. But then what they found is that bacteria that had gone into the light medium, okay, produced an intermediate. In other words, they took the bacteria into the heavy, and then some of that bacteria went in the first test tube, which is only heavy. Test tube 2 was bacteria that was in the heavy medium, then in the light medium, and what they found is that DNA that was duplicated was a combination of the two types of DNA. And of course, the third group was DNA grown in just the light medium only. Or I should say, no, I'm sorry, I misspoke. Bacteria in the heavy, then the light, and in that third test tube, they got a combination of that intermediate and in light only. So they were now investigating what was going on. All right, so that led them down a path to be thinking, okay, why would the DNA strands be different, and what their process was, or their idea was. Something must be going on, and because the mindset was that whatever the DNA was, like if it was DNA was replicated, you separate it out and then make copies. And the mindset was, all right, then the two original DNA strands are put back together, and then the two newer strands will be put together, and you go forward. But that was not what was happening. Okay, what they were learning is the fact that when DNA is replicated, the original DNA is matched with its copied DNA and then put back together. It's no longer just the two pairs. So essentially, if these are the two original DNA strands and we make copies of those, then this one, oh, I'm moving the wrong hand. Oh, I need to be over here more. Okay, then this one goes with its copied, and this one goes with its copied, and now we have an old and new paired together. So that was their conclusion on that process, is that DNA is replicated in a semi-conservative process, meaning the original DNA strand is matched back together with its copied, the new strand, not the two old ones put back together and the two young ones put together, but an old and a young together. All right, so our last scientist, Fred Sanger, come up with a process of growing, essentially, DNA strands. In his process, prior to him doing this, like with everything else, not sure how to do it, but the way they had was expensive and long. And if you're going to study DNA and everything about it, you want to be able to do so in a quick and efficient manner. So he came up with a system that ended up being used, as I mentioned, as a part of the sequencing, gene sequencing, the genome project. So we're going to take a look at that process. Alright, so it's the idea of step one was simply to determine what DNA sequence that you wanted to replicate, and then you've got your beakers with your C, G, A, and T's in it. Run that solution, run it through that solution, and then then you get your DNA strand being made. So then you can, and based on what nucleotides are matched, then you can then sequence what the original strand was. So that's the idea of, or Sanger's contribution to this process of understanding what is DNA. Well, that's our video on experiments part 2. Next time we come together, we're going to be talking about the structure of DNA. We'll see you then.